We're going to go over transcription in this lecture. Okay, let's take a look at transcription in bacteria. So starting transcription initiation stage, we need a little bit of help for the polymerase to bind to the promoter here in yellow, the start of the gene on the DNA. And so there's a little protein here called the sigma factor, and it's just named after a Greek letter. And so when the RNA polymerase goes to bind to the promoter on the DNA, the sigma factor will help it bind to the promoter. And then when the RNA polymerase gets going and starts transcribing, then the sigma factor just falls off because its job is done. Okay, so sigma factor just helps the RNA polymerase to bind to the promoter. Okay, and then this is just looking at the promoter in the DNA. And you can see there's lots of T's and A's there. And the RNA polymerase will recognize the sequence and know that, hey, that's the start of this operon or maybe this gene, and that's where I'm going to bind and then start transcribing. Okay, so the RNA polymerase is going to be reading the template DNA strand, which in this picture here is going to be the light purple. And it's going to read in the 3' to 5' direction. So we're going to be going that way. So we'll fill in our blank here. So we're always going to read in the 3' to 5' direction. And that's just like we talked about in the DNA replication. And then we're always going to synthesize the new messenger RNA that we're making here. So here's our 5' end, and then this here would be our 3' end that we're adding the RNA nucleotides to. So we're going to synthesize the messenger RNA in the 5' to 3' direction. So I would definitely know these directions for the quiz and the exam because that's a pretty easy test question. Okay, and then also if we just draw the DNA here, and if we're reading it in the 3 to 5' direction, and then here's our messenger RNA that we're making, and that'll be in the 5 to 3' direction that we're making it. Notice that the DNA template strand and the messenger RNA are going to be anti-parallel, so they're going to be running in the opposite directions. And they're also going to be complementary to each other. So A's and U's, and you've got T and A, and G's and C's that will go together. Okay, and then this just shows everything together. So you can see that starting or initiating the transcription, we're going to have the sigma factor, that little protein that's going to help the RNA polymerase bind to the promoter. The RNA polymerase will break the hydrogen bonds between the two DNA strands, separate the template strands, so it can use that to make the messenger RNA. And then it'll read the template strand in the 3 to 5' direction, and then make the messenger RNA in the 5 to 3' direction. And then it'll just run along the DNA until it gets to the terminator sequence, the end of the gene, and then the RNA polymerase will fall off, and that'll be the end of the messenger RNA that you make. Okay, and then this is just showing the RNA polymerase getting to the terminator sequence, and then it just falls off and releases the messenger RNA. Okay, and then transcription in eukaryotic cells is fairly similar, just a few differences. So here's the promoter in the DNA, and the RNA polymerase will recognize that sequence of T's and A's. They call it the Tata box in eukaryotes. And what's going to help the RNA polymerase bind to the promoter, instead of the sigma factor, we're going to use transcription factor. So they're just a little bit different proteins there. So they'll help the RNA polymerase bind to the promoter. RNA polymerase will break the hydrogen bonds and then use that template strand, read that, and then make the complementary messenger RNA to go along with that. Okay, and then this is just showing you again RNA polymerase going ahead and using the template strand, and it will be reading the template strand in the 3' to 5' direction, so it would be going this way. And then the messenger RNA that's being made, 5' end would be there, and then the 3' end would be here that you're adding the new RNA nucleotides to. And then when it gets to this terminator sequence, that's the end of the gene there, and then the RNA polymerase will just fall off and then release the messenger RNA. Okay, another big difference with the eukaryotes from the prokaryotes is that the messenger RNA that's produced, we have to do 3 things to it. We have to modify it before it's ready to translate. So the 3 things that we need to do to the messenger RNA before it actually leaves the nucleus and goes to the cytoplasm for translation, we have to do things to both ends and then something to the middle. So we're going to add a 5' cap to the 5' end of the messenger RNA. And then we're going to add a poly A tail, so a bunch of A's to the 3' end. And then in the middle, we're going to do the RNA splicing. So we're going to cut out the introns and then splice the exons back together. All right, so this picture on the bottom, you can see the 5' cap that we put on. And then on the 3' end, here's our poly A tail. So we put a bunch of A's on the 3' end. Okay, so the reason that we do the 5' cap and the poly A tail, there's 3 reasons here, and these would be good to know for the quiz and the exams, be good test questions. So they're going to help the messenger RNA exit the nucleus. So if the cap and the tail are not on the messenger RNA, the nuclear pores, little holes in the nuclear envelope, will not let the messenger RNA out because it knows it's not ready to leave yet. So you got to have the cap and the tail on before you can leave the nucleus. And then also it's going to protect the messenger RNA from being broken down by enzymes when it's in the cytoplasm. So by having the cap and the tail on, the half-life of the messenger RNA out in the cytoplasm will be about 10 hours. And so the ribosomes can keep hopping on and keep translating that messenger RNA, and you can make lots of copies of that protein. But if you didn't have the cap or the tail on, the half-life of the messenger RNA is only about 2 minutes. So you wouldn't have very long to make the protein. So it's good we have the cap and the tail on. It gives you more time to make lots of copies of that protein. Okay, and then the last thing, the reason that we have the 5' cap is it's going to help the ribosome recognize which end is the 5' end, because that's the end that we need to start translating from. Okay, so I would know those 3 things for the quiz and the exam because that would be pretty easy to put on a test. Okay, so definitely know those things. All right, so that's the cap and the tail. And then the third modification we're going to do the messenger RNA in the eukaryotes is RNA splicing. So the exons are the expressed sequences. That's how they got their name, exon. So these are the parts of the messenger RNA that we're going to translate and make into the protein. And then the introns are the intervening sequences that we're going to cut out and not use to make the protein. Okay, and what's going to do the splicing would be the spliceosomes. So here's a picture of a spliceosome, this whole big purple thing, and you can see that it's cutting out this lighter reddish pink intron, and then it's going to splice together these red exons together. So a spliceosome is just made out of this red small nuclear RNA, and it's just named that because it's small and found in the nucleus. And then we've got this purple protein, and so this whole thing is called a SNRP, which I really like that name. That's a cool name, SNRP. And then we just have some other proteins combined with the SNRPs, and that makes up a spliceosome. So spliceosomes will do the RNA splicing and cut out the introns and splice the exons back together. Okay, and this is just a summary of the 3 modifications to the eukaryotic messenger RNA before it's ready to translate. And so all this will be happening in the nucleus. So here's our DNA. Here's our pre-messenger RNA before we do anything to it. And then here's after we've done the 3 modifications. So we're going to modify both ends. We put a 5' cap, poly A tail, and the 3' end. And then in the middle, we're going to do our RNA splicing where we cut out the introns. All right, let's fill out our table here. This would be good to study. So let's summarize the differences between bacteria and eukaryotes in transcription. So where it occurs in bacteria would be the cytoplasm because they don't have a nucleus. And then eukaryotes, transcription will happen in their nucleus because that's where the DNA is. And then the next one, what helps the RNA polymerase initiate or start transcription. So what's going to help it bind to the promoter. So in bacteria, the protein is called a sigma factor. And in eukaryotes, not very original name, the proteins are just called transcription factors. So they will help the RNA polymerase bind to the promoter and start transcription. And in bacteria, do they have monocistronic or polycistronic messenger RNA? And they can actually have both, but they do have probably mostly polycistronic messenger RNA because they do have a lot of operons. And then eukaryotes will have the monocistronic messenger RNA. Okay, and messenger RNA processing, do we have to do a 5' cap, poly A tail, RNA splicing in the bacteria? And hopefully you said no. So the bacteria, as soon as they make their messenger RNA, it's ready to translate right away. But the eukaryotes, they do have to do all 3 of those things.